In this video, we're going to talk about hydrogen bonds. Hydrogen bonds occur when two electronegative atoms, like oxygen and nitrogen, share a hydrogen. Unlike covalent bonds, hydrogen bonds are weak and easily broken. This is important in biotechnology, especially when we work with DNA. Hydrogen bonds can be a little confusing because most structures don't show them. We see them in structures that were solved by NMR, but structures that were solved by other methods don't show them. So we have to use our knowledge of chemistry to predict where they're likely to be, and look for cases where oxygens and nitrogens are close together. Sometimes this can be a little bit confusing because some students can get hydrogen bonds mixed up with ionic bonds. To clarify this, hydrogen bonds, remember, occur when a hydrogen is shared between an oxygen and nitrogen, or two oxygens, or two nitrogens. Ionic bonds, however, only occur when the oxygen has a negative charge and the nitrogen has a positive charge. Now we're going to look for a hydrogen bond in the structure. So I open up the residues, I change to spacefill, and I'm going to turn the structure around until I can find a hydrogen bond. So I'm looking for a hydrogen between an oxygen and a nitrogen, or two oxygens or two nitrogens. I zoom in a little bit by spreading my fingers apart, and I can see there's a hydrogen right there in the middle. Now I want to figure out what those residues are. So I change to residue coloring to help me identify them, and I open up the color key. The blue residue is a lysine, I can see, and the gray one is a glycine. Now what I want to do is I'm going to close the color key, and I'm going to look for G's and K's and touch each one in the sequence until I highlight the ones that have the hydrogen bond. There's the glycine. Now I'm going to look at K's and see where I can find the lysine. Okay, now that I've found the lysine, I'm going to hide everything else. I'm going to change the coloring style back to element, and I'm going to turn this so I get a little bit better view of the bond. So you see that hydrogen right there between the oxygen and nitrogen. Okay. Now what I want to do is figure out where these are located in the chain. So I touch each of the residues, and I see the number appear, and I get the chain location.